17.4 Monohybrid inheritance. Don't forget to go back to the objectives to make sure that you're done studying correctly. What's inheritance? Inheritance is passing the characteristics from one generation to the next one, which means from parents to their offspring. What can you see in this photo? You see the father and the mother. And now you can see the son has a mix of characteristics. Some of them inherited or taken from the father, and some of them are inherited or taken from the mother. There are about 20,000 human genes carried on the 23 pairs of chromosomes. So again, this is the karyotype or the microscopic image of our chromosomes. Each one of them carries a lot of genes. That's why we have around 20,000 genes carried on those chromosomes. What are homologous chromosomes again? These are the pair of chromosomes carrying the same genes at the same positions. So why do we have two? Because we have one from the father and one from the mother, a paternal copy and a maternal copy. So we have two copies or two forms of the same gene carried in the same position on the chromosome. And we learned before that different forms of the same gene are called alleles. How do we refer to genes when we're writing a genotype? What's a genotype? It's the genetic makeup or the composition of the gene that appears on us. So what's inside the genetic makeup that we don't necessarily see all of it is called the genotype. We refer to it by letters. Either those letters can be capital or lowercase letters. Capital letters usually refer to the stronger genes or the stronger alleles, the ones that will appear in our phenotype or in our physical appearance, while the weaker ones that do not necessarily show in our physical appearance, we call them recessive. Let's look here at this example. So all of those individuals or all of those animals have the fur color gene, but the different alleles here or the different forms of this gene are either spotted fur or black fur. If you look here, you will see that the father has spotted fur while the mother has only black fur. But what do you see in the offspring? You see in the offspring only the spotted fur. So again, which fur allele is stronger? Actually, the spotted one. So we call the strong one the dominant allele. Dominant means that it can hide the other allele totally. Though all of the offspring inherited one copy from the father and one copy from the mother, so they must have in their genotype or in their copies of the gene the black fur, yet they only show in their physical appearance or in their observable features only the spotted fur. So this means that the spotted fur allele is dominant or stronger than the black fur allele. That's why it's totally hidden and you cannot see it in the outside or in the phenotype. So recessive genes might be in the genotype, but you will only see the dominant ones. And the recessive ones or the weak ones don't appear in the phenotype. So here, when you have a spotted coat allele and a spotted coat allele, you will necessarily have a spotted offspring. If you have spotted coat and black coat, you will receive or you will have also spotted coat. Why? Because as we agreed, the spotted is dominant over the only black. But if you look here in the F2, you will see the black appearing again. That's why you can see the recessive allele only when you have both of the alleles in the genotype recessive. Let's take a look here so we can understand it better. If you look here, you will see the genotype of those individuals. What's a genotype again? It is the genetic makeup in terms of alleles. And we agreed before that we refer to genes as letters. So let's take a look here. We have a father with blue eyes and a mother with brown eyes. So if you take a look, you will see that the blue is written in lowercase letters, which means that it is recessive or weak. And that's why her eyes appear to be blue, because she has both of her alleles in the recessive form. So that's why you can see now the recessive allele in the genotype. While the father here has one strong or dominant allele and one weak or recessive allele. So that's why his eye color follows the dominant one and they appear brown. So again, the phenotype is the physical appearance. Here the physical appearance appears blue, while here it's brown, because here both of the alleles in his genotype are blue, while here it is a mix, the brown and the blue, so the one appears in the phenotype is the strong one. So again, what's the phenotype? It's the observable features or the things or the feature that you can see on the outside. So it's influenced by the genotype and sometimes with the environment. Not in the case of the eye color, of course, but for example, in the case of height, in the case of weight, it's also affected by our environment or our behavior. So genotype consists of a dominant allele that's expressed in capital letters, as you can see here. So this one is the dominant allele. And a recessive allele that's expressed in the lowercase letter, and it's always hidden under the dominant one. So if you have a dominant allele and a recessive allele, the one appearing in the phenotype will always be the dominant allele only. Homozygous. What's homozygous genotype? Homozygous genotype when you have both alleles the same. So you can have a homozygous recessive genotype, and this is the only case where you will see the recessive allele in the phenotype or as an observable feature. Or you can also have, that's not in this example, a homozygous dominant genotype. What does this mean? This means that both alleles are the same, but they are both dominant. And we have another type of genotype called the heterozygous. What's heterozygous? It's the mix. When you have one dominant allele and one recessive allele. And I don't care which is coming from where, but you have to always write the dominant allele first in the genotype. Also, you have to make sure that for every gene, you give only one letter. If those were green eyes, I don't care. They will take the letter B also. Why? Because we follow the letter of the dominant allele. Okay, so those individuals back here, they had 2 copies of the same gene. Why do I see them giving their offspring only 1 copy? Because when gamete cells are made, division of the alleles happen, because we agreed before that the gamete cells are haploid. What does haploid mean? It means that they have half the number of chromosomes, which means that in our gamete cells, we have only 1 copy of each gene or 1 allele. So the female egg will always have only 1 copy of the 2 alleles that the mother has. And also the male sperm will always have just 1 copy from the 2 copies that the original male has. Monohybrid cross. What does this mean? This means that we are only considering the inheritance of one gene. Do we actually in real life inherit one gene? Actually, no. We said that we have around 20,000 genes. So we will never inherit one gene. But this is a way to trace the inheritance of only one gene. And of course, it's not 100% accurate, but it gives us an image about how we inherit a specific gene. Let's take a look at the leopard's example. Now let's take a look at the fur color of all of the leopards. So we chose the letter B for the dominant allele. And of course, if you chose a letter for the gene, you have to put the same letter for both dominant and recessive alleles, even if they don't start with the same letter. This has nothing to do with the initial. This is related to same gene must have the same letter. So here we go, B capital and B lowercase. So the spotted mother, for example, is heterozygous. What does heterozygous mean again? It means that it has both the dominant and the recessive alleles. And the father also was heterozygous. Now, can you tell me, what does this mean? This means that their phenotype, both of them, is spotted, because we agreed before that we will always see the dominant allele in the phenotype. Now, making gametes, we have to separate both alleles because the gamete cells are haploid. They have only half the genes. They have only one allele of the gene. So here we go, gamete, those are egg cells and those are sperm cells. Now, in order to make offspring from this family, you need to take one egg cell and join it with a sperm cell. So the first option or the first individual in the generation will be B capital. So this means that it is homozygous because they are both the same, and dominant because they are both capital, written in capital letters. So this genotype is said to be homozygous dominant. And what will be the phenotype or the physical appearance? Of course, it's gonna be spotted. Now, what is the second possibility or probability? If this egg cell joined with the second option of sperm cells for this gene, it's gonna be B capital and B lowercase. So now we have one dominant allele and one recessive allele. What does dominant and recessive make together? They make a heterozygous genotype or a mixed genotype. What will appear in the phenotype will be only the trait of the dominant allele. So we will see again a spotted leopard. Now, both options of this egg cell are done. Let's see the options of the second egg cell. It's gonna be lowercase b joining with the capital B, which means that, again, it will form a heterozygous individual. Why didn't I write the lowercase b first? Because the dominant allele must be written first all the time. And now the last option will be this egg cell joining with this sperm cell, and you will get two recessive alleles, which is homozygous recessive. Please make sure while writing the genotype to mention, in case of homozygous, either it's dominant or recessive. You can never write just homozygous. You have to mention homozygous dominant or recessive. And the phenotype here is gonna be different. It will follow the recessive allele. How? Because both alleles are recessive, so there is no other source for the phenotype. That's why the phenotype will appear black. We can use the monohybrid cross, as I showed you previously, or we can use the Punnett square. It is a genetic diagram also, but it is more practical and more organized to show us or to make us know all the possible phenotypes and genotypes from a specific cross for one gene only. So it's a neat way that will give us the full image in an organized way. How does it work? It's a box that's divided into 4 squares, which represents 1/4 of the percentage. What does this mean? This means that each square here represents 25%. 25%, which is 1/4 of the probabilities of this family for this gene only. So if we put the gametes here, just like the previous example, we will put the male gametes, the sperms, and the female gametes, the eggs, and then we will join this egg with this sperm and see the outcome. Let's revise again. This is homozygous dominant. Both are the same and dominant allele and following the dominant trait, which is the spotted fur. And then here, as you see, this is the recessive gamete or the recessive allele that will join with the dominant one. You will get a heterozygous genotype, and it will follow the dominant allele to become a spotted leopard. And now you get the same, while here you get both recessive, because you will take one egg with one sperm and both happen to be recessive, or both happen to carry the recessive allele of this gene. So it will appear black. So let's do the percentages. The genotypic percentages will be 1 homozygous dominant, which holds 25%. So now the homozygous dominant will be equal to 25% of the offspring. What about the heterozygous here? If each one of them is 25%, or each square, is 25%, so the heterozygous will be equal to 25 plus 25, which is 50%. What about the homozygous recessive? It will be equal to 1 square, which is 25% only. What about the ratio? How do we calculate it? Again, each square represents 1 out of 4. So if we're going to make the ratio, we will do first the 3 categories that we have, which are homozygous dominant to heterozygous to homozygous recessive, which will be only 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. So these are the genotypic ratios. Are they the same as the phenotypic ratios? What's phenotype again? It's the observable features or the physical appearance. Here I see the options are spotted to black, as you can see. So what's the ratio of the spotted offspring? We have 1, 2, 3. So it's going to be 3. And what's the ratio of the black offspring? It's going to be 1. So sometimes the phenotypic ratios are quite different than the genotypic ratios. Genotype is the genetic makeup, while the phenotype is the physical appearance. Okay. So, possibilities and predictions. We have possibilities. If the 2 individuals are homozygous and are crossed together, of course, the offspring will be homozygous for those alleles. We call them pure breeding. Have you ever heard the term a pure dog? What does this mean? It means that all of the characteristics are surely inherited through all the generations the same way. And the only way to make sure that this happens is that both alleles are the same, so that no other features will appear in the offspring. So all of the alleles of this dog are similar. Both alleles of the same gene are similar. That's why we are sure that the offspring of this dog will contain the same feature as is, the same physical appearance, the same phenotype. While if we have heterozygous parents, as here, this means that this dog or this animal, for example, I'm just talking about the example I said earlier, got crossed with another dog that carries different features. So now there is a possibility in the alleles of the offspring that they have different features that are not favorable. So this is what we call not pure breeding. And that's why the offspring of the dog for the second generation will not contain the same favorable features. So again, if both alleles are homozygous for the trait, this means that it's a pure breeding for this trait. So this means that the offspring will surely carry the same characteristic as is. We call this pure breeding. While if parents were heterozygous, this means that we can have mixture of the phenotypes of this trait in their offspring. And this is what we call not a pure breeding. And usually, if two heterozygous individuals were crossed, they will produce the phenotypic ratio as 3:1. 3 of their offspring will carry the dominant allele of phenotype, while 1 of the offspring will carry the recessive one. Okay, let's take a look here to make sure that you understand the point of the difference between the phenotypic and the genotypic ratios. Here you have a heterozygous individual crossed with another heterozygous individual. Now you will have in the genotypic ratio homozygous dominant 1, heterozygous 2, and homozygous recessive 1. And the phenotypes will be yellow, 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 3, while 1 green, 1. And here the yellow is the dominant, as you can see. Now, if you have two pure individuals, but not the same, one of them is homozygous dominant while the other is homozygous recessive, were crossed together, the genotypic and the phenotypic ratios will still be the same. Why? Because now you have all of the individuals will be heterozygous and all of them will carry the phenotype of the dominant allele. If you look here, if you have one heterozygous crossed with one homozygous recessive, now we put the gametes and we start doing or using our Punnett square. We will have two heterozygous individuals and two homozygous recessive individuals, which will be the ratio of 1:1 as a genotypic ratio and also as a phenotypic ratio. What's a pedigree then? Those were the ways we predict what could be the outcome of this crossing, while the pedigree is explaining to you or showing you how families inherit a specific trait or what happened in real life. What happened in this family specifically? What happened through or from this cross? So now let's see. We have different generations in the pedigree. First, you have to know that males are represented as squares while females as circles. So whenever you look at a pedigree and you see a circle, this is a female. While when you look at a square, this is a male. The horizontal line connecting the square with the circle here, this is a marriage line or the crossing line. And now this vertical line represents offspring. Okay, so the vertical line connects offspring to their parents. And when you see a shaded square or circle, this means that this individual shows the phenotype of the trait we're following. For example, if I am following the inheritance of blue eyes in a certain family, All the shaded individuals have blue eyes. This means that I am following the trait of blue eyes in the phenotype. So the pedigree shows me necessarily the phenotypes, and sometimes it helps me to predict or to know the genotypes when we follow the whole diagram. Okay, so what about this horizontal line? Is it a marriage line? No, it's not connecting a specific circle with a specific square like here. So these are siblings. Think of this as a bracket. It's connecting different individuals coming from this family, but they are not married. It's just an extension of the vertical line. So here you have 4 siblings coming from this family, from those father and mother, and this boy married this girl from outside of the family, and this girl married this boy from outside of the family. Okay, so now I know that the shaded ones show the phenotype, and the non-shaded ones do not show the phenotype. What about the half-shaded ones? The half-shaded ones are carriers. What does carriers mean? This means that we know for sure that they are heterozygous for the trait. They are not showing it in their phenotype, but we know that they carry it. Okay, so again, the shaded ones are showing the trait in their phenotype. The non-shaded ones, they do not show the trait in their phenotype. Also, the half-shaded ones, they do not show it in their phenotype, but we know that they are heterozygous for it. So we know that they are carriers of the recessive gene that will show in the phenotype if both alleles were recessive. Now, there are some other symbols that are not showing in this pedigree, like if you cross out a circle or a square, this means that this individual is deceased or dead. If you have 2 coming out from the same point, this means that they are twins. They were born at the same time. Individuals between brackets, like here, means that they are adopted. So having them in the family pedigree, if they have the phenotype or not, will not help us predict the genotypes of the whole family. What about the triangle? If you see a triangle, this is a miscarried baby or a baby that was not born alive. We call the generations with the Roman numbers. So this is generation I, double I, triple I, IV. Now, if you look here, the first generation, they have 4 offspring, right? But if I ask you how many individuals in generation double I, you have to count everybody. So again, if I say how many individuals in double I, you will count 1, 2, 3, 4, 5, 6. So this means that I have 6 individuals in this generation. But they are not all coming from this couple. So if I ask you how many of generation 2 are the offspring of generation 1, you will count only the offspring of generation 1, which is going to be 1, 2, 3, and 4. Okay, so this is how we read a pedigree. Sex determination. How is the sex of the newborn or the sex of the fetus decided? It's decided by the sex chromosomes. So how many sex chromosomes do we have? As we agreed before, we have 2, X and Y. The genotype of a female is XX, while the genotype of a male is XY. But didn't we agree that a gene has to be called with the same letter? Yes, but this is not a gene, it's a full chromosome. And in your questions, you will only see this case with the sex chromosomes. We will not be asking you about the inheritance of a full chromosome, except only in the case of sex chromosomes. Something else you need to know is that the X chromosome is much larger in mammals and it carries a lot more genes than the Y chromosome. And that's for obvious reasons, because the X chromosome is present in both genders, while the Y chromosome is only present in males. So the Y chromosome is only responsible for gender determination, while the X chromosome contains other somatic genes or genes responsible for other traits. Therefore, male's gametes are the ones that decide the gender or the sex of the baby. So here you have the father has an X chromosome and a Y chromosome, while the mother, the only options she can give to her baby are X's. So now when you divide the alleles or when you divide the options of the father, you have X chromosome and Y chromosome. While when you divide the chromosomes or when you have two copies of the chromosome, each copy goes to a gamete cell, you will see X and X in the case of the mother. Doing the monohybrid cross, you will see that the daughter will always inherit an X from her father and an X from her mother, while the son will always inherit the Y from his father and the X from his mother. Can the boy inherit the X of his father? Of course not, because the mother will never be able to give him a Y. So, if the baby got an X from the father, this means that this baby will turn out to be a daughter, because she will necessarily receive another X from the mom. So again, only the sperm cells are responsible for deciding either this family will have a son or a daughter, because only sperm cells have the two options, while the gamete cells of the mother, the X cells, will only give X's. These are the only options the mothers have. Okay, let's answer some questions together. The family tree shows the inheritance of the ability to taste a certain substance. The allele for the ability to taste this substance is dominant to the allele of the inability to taste the substance. So here you have that a shaded one is a tester male and a shaded circle is a tester female, and non-tasters in males are non-shaded and non-tasters in females are non-shaded. Now, he told us in the question that the ability to taste this substance is dominant over the inability. So accordingly, we will know that anybody with the inability are recessive, carrying the recessive characteristic, right? So this one must be recessive. Both alleles are recessive. And here both alleles are recessive, both alleles are recessive, both alleles recessive. So this is how we guess the genotype. It's easy to know the homozygous recessive ones because they are the ones that are showing the recessive trait. The dominant ones or the ones showing the dominant trait are the tricky ones because they can be either homozygous dominant or they can be heterozygous. So how will I know? I will look at the offspring. If I see one of them homozygous recessive, this means that they must inherit another recessive allele from the other partner. So this means that this individual must be heterozygous in order to be able to provide the other recessive allele to her daughter. Again, so we know for sure that this father is having both of the recessive alleles, homozygous recessive. Why? Because in his phenotype, I can see the recessive trait. So this means that he must be homozygous recessive because the recessive trait only appears in the case of homozygous recessive. But the mother here is tricky because in the case of heterozygous or homozygous dominant, I will still see the phenotype of the dominant trait. How would I know? If I look at the offspring and I see one of them showing in his or her phenotype the recessive trait, so this means again that this person is homozygous recessive. And in order to have a homozygous recessive offspring, we must have one recessive allele from the father and one recessive allele from the mother. So this means that the mother must have a recessive allele, which means that this mother is heterozygous. So now we know that the mother here is heterozygous. And accordingly, we know that this person here is heterozygous too. Why? Because the father here is homozygous recessive. Let's see the question that he's asking. What is the chance of the second child of Peter and Hannah being non-tasters? For a person to be a non-taster, this person has to be homozygous recessive. Let's do the Punnett square. We know that Peter is heterozygous, as we agreed before, and Hannah is homozygous recessive, as we agreed before. So if we're going to draw a Punnett square, looking like this, we have the 4 options. I'm not very good at drawing. So here we go. We will have one of Peter's gametes here. Let's choose the dominant one. And the other gamete will be the recessive one. And Hannah's gametes are only recessive. Let's do the first option. So the first son will be heterozygous, taking this gamete with this gamete. And the second individual will be homozygous recessive. The third one will be heterozygous too. And the last one will be homozygous recessive. So if we look at the Punnett square here, we can easily know. Now, we want to know the non-tasters. So, which ones are non-tasters? We agreed that non-tasters are only the homozygous recessive ones. So, this is half the number, which is 1 in 2. So this is how we answer a genetics question. We have to make sure that we understand the phenotype and the genotype and their indicators, and we have to make sure that we draw our Punnett squares. Okay, what are the chromosomes for the 2 sexes in human beings? We have only XX for female and XY for male. In some animals, B is the allele for normal skin. And if you see the question writing the letter as a capital 1, this means that this is the dominant allele. And lowercase b is the allele for spotted skin. So which one is dominant? The normal skin, which is without spots. A pair of these animals has 37 offspring. If you look at the ratio, you will see it almost 1-to-1. It will never be exactly 1-to-1, of course. What are the most likely genotypes of the parents? So, of course, since we have a mix, so this means that they will never be homozygous dominant crossed together or homozygous recessive crossed together. So we're going to eliminate those 2 options. These are not the options, of course. And then we will take the other 2 and we will do their Punnett squares. Let's draw the Punnett squares now. So here we go. Let's go for the first option. Here is the square. We go with the 4 squares inside. As I told you before, I'm not very good at drawing. Now we will do the gametes. So we have B capital, lowercase b, and B capital again, lowercase b. So the first option of the offspring will be homozygous dominant. So homozygous dominant, which is without spots. And now, again, the second option, this one will be heterozygous one from here and one from here, and this one will be heterozygous, one from here and one from here, while the last one will be homozygous recessive. So we have the ratio here, the phenotypic ratio 3:1, which is definitely not the ratio mentioned in the question, because the question's ratio is almost 1:1. So again, B also is excluded. So the right answer is C. If you do the Punnett square, you will find out that the phenotypic ratio will be 1:1. So please always do the Punnett square when you're confused between two options. Cystic fibrosis is an inherited disease that occurs when an individual is homozygous for a recessive allele. If parents are both heterozygous for the characteristic, what is the probability that their first child will have cystic fibrosis? So again, we can do the Punnett square. But we know that if two heterozygous individuals were crossed together, there will be 25% that the recessive trait appears in the phenotype of their individuals, because the ratio will always be 3:1. So it's gonna be 25%. What determines the sex of the baby? The father's chromosomes, of course, because we said that the sperm cells are the only ones that can carry X or Y, while the mother will always give the offspring the X chromosome. When white flowered pea plants were crossed with red flowered pea plants, the offspring in F1 were all red flowered. So this means that they were both homozygous. So the white flowers were homozygous recessive, while the red flowers were homozygous dominant. So this means that all of the F1 generation are heterozygous, taking one allele from the red flower and one allele from the white flower. So if you cross two heterozygous together, you will get the ratio of 3:1 again. So why did you see white flowers in the F2 generation or in the third generation? That's because the allele of the red flower is dominant and the F1 plants are heterozygous. Which sex chromosomes are present in all mature human sperm cells? It can be either X or Y. Why didn't I say both X and Y? Because both means that the sperm cells are diploid, or that they have both alleles of a specific gene or both copies of a chromosome, and this is not the case. We said that the sperm cells are gamete cells and they are haploid, carrying only one copy of each chromosome and accordingly one allele from each gene. The chart shows the inheritance of fur color in small mammals. If the alleles for white fur is dominant, which animals must be heterozygous for the gene controlling the color? So now we know that the black ones are homozygous recessive, right? So we will only be confused between A and C, because B are definitely homozygous recessive because they are showing the phenotype of the recessive allele. So now we're looking at A and C. Which ones will be necessarily heterozygous? Of course it's C, because one of the parents here is homozygous recessive and the other parent is showing the dominant allele, which means that it's either heterozygous or homozygous dominant. So this means that this one is definitely heterozygous. So the answer will be C. The diagram shows a potato tuber that developed from the stem of the parent potato plant. 3 shoots are starting to grow from the tuber. How do the genotypes of the shoot compare to the genotypes of the parent? Of course, they are both individual because this is a type of asexual reproduction. So it's identical to the shoots, identical to the shoots. Figure 6.2 shows a family tree. So this means that what we're looking at is a pedigree, in which the inherited condition beta thalassemia occurs. Beta thalassemia is caused by a recessive allele. That's why it's represented by a lowercase p. It results in the formation of hemoglobin that carries less oxygen than normal hemoglobin. So now we know that anyone that shows symptoms of beta thalassemia must have a homozygous recessive genotype. So now he's asking you, child 2 of this family has beta thalassemia. So what do you think is the genotype of child 2? So the answer will be that child 2 will be homozygous recessive because he or she has beta thalassemia. Now he's asking you about child 1. What do you think will be the phenotype of child 1? They will produce normal hemoglobin. Why? Oh, he or she will produce normal hemoglobin. Why? Because their genotype is homozygous dominant, and in the dominant case, a person does not show any symptoms. Then the question is asking you about the genotype of the father and the mother. And this is gonna be heterozygous. How did I know that they were heterozygous? Because, as we said before, for an individual to carry the homozygous recessive genotype, this indicates that both parents must carry the recessive alleles for the person to collect one recessive allele from each parent. So now he's asking you lastly to complete the diagram, and this is what we did in the beginning. Okay, let's see the final part of this question. He's asking you state which 2 people in this family are heterozygous for the condition. Of course, these are the father and the mother. Now, 6.2 shows the sequence that occurred during sexual reproduction. So sperm, egg. So again, this is a question that's collecting many topics together. Sexual reproduction with inheritance. Please make sure that you know how to connect the topics together. This is a very important assessment objective, as we discussed in the beginning of the year. Some humans have the ability to roll their tongue, and some cannot roll their tongue. This characteristic is controlled by genes. You can see here boy A and boy B. These are the differences between the phenotypes. The allele for the tongue rolling, T capital, is dominant over the allele of non-tongue rolling, which is lowercase t. 6.3, remember when I told you it's very important to read the question carefully and know which diagram am I looking at. As you see here, this is a very important example. Now you will answer based on a specific diagram. 6.3 shows a family tree, which is a pedigree again, for this characteristic. Individual 1, here we go, and his partner are both heterozygous for tongue rolling. So we know now that individual 1 has the genotype that's heterozygous and the partner is the same. Now he's asking you complete table 6.1 by inserting the genotypes of the numbered individuals in figure 6.3. So please make sure you read the names of the figures to know where are you looking. So now he's asking you about individual 1. Individual 1, we agreed since he told us that he's heterozygous, so we know that it's heterozygous. What about individual 3? What do you think? Is she heterozygous or not? Why can't I say that she is homozygous dominant? Again, we will look at the offspring of individual 3. Now we have one of her offspring That is homozygous recessive because this female is non-tongue rolling. So this means that both parents must have the recessive allele. So again, individual 3 will be heterozygous. And then finally, for the same reason, individual 4 is heterozygous. So again, individual 1 heterozygous, 3 heterozygous, and 4 is heterozygous too. Individual 2 in figure 6.3 is heterozygous for tongue rolling. He marries a woman who cannot roll her tongue. If she cannot roll her tongue, this means that she is homozygous recessive. Both alleles recessive and similar. State all the possible genotypes of their children and predict the ratio of the phenotypes. In this case, we must draw a Punnett square. So we will draw the Punnett square and put the 4 possibilities. And then we put the gametes. We knew that individual 1 was heterozygous. So 1 gamete, the other gamete, and he married a non-rolling, which means that she is homozygous recessive. There we go. So the first probability will be heterozygous, and the phenotype will be rolling or can roll his or her tongue, and this one will be homozygous recessive, which is non-rolling, and then heterozygous, and since this individual has the dominant allele, so they are going to be rolling, while here the last one will be non-rolling. So let's take a look at the answer. It's gonna be, the possible genotypes will be heterozygous or homozygous recessive with the ratio of 1 to 1. Let's look at another question. Throughout the world, there are almost equal numbers of female and male baby borns. The sex of the baby is determined by the sex chromosomes. State the sex chromosomes present in female and male. So here we go. Females are always XX while males are always XY. Complete the genetic diagram to show the inheritance of sex chromosomes in humans. So the female will have the sex XX and the male will have the sex chromosomes XY. And the gametes of the female will only be X and X, or will carry only the X chromosomes, and the male can carry an X chromosome or Y chromosome on his sperms. Now, since this diagram is a monohybrid cross, so we will do the monohybrid cross lines. We will join the eggs and sperms by turn, and you will continue with the same way and so on. So you will finally get 2 females and 2 males, 50-50. Thank you, and I hope this video helps you study.